This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 71, recorded on May 9th, 2014. Hi, everybody. I'm Vincent Racaniello, and with me is a smiling Dickson Despommier.
Good afternoon, Vincent.
Why are you smiling?
It's not a great day out there, but I'm smiling anyway because I'm just in a good mood. You can smile for almost no reason at all.
You actually are mostly in a good mood.
That's true. I happen to be a very positively oriented person.
Do you think I'm mostly in a good mood? You can be honest.
You know, it's tough to judge your mood when you come in the morning. I'm being honest, but I find you pleasantly oriented most of the time.
You don't have to.
It depends on what's going on. You know, I mean, if there's a lot of things going on, you have to take trips and stuff like this. I think you exhibit a harried attitude, which I would too, by the way.
Yeah, I get harried. But
harried is the best answer there.
I'm not in a bad mood, but I yell at you a lot, I know, because I'm
but
impatient with you.
You're just teasing. So let's talk about the weather for a moment. Let's look out the window. You know what?
It's like soup.
New Jersey's gone.
It's like soup.
There is no New Jersey out the window. In fact, there's nothing out the window except this looks like we're in the middle of a cloud.
Yeah.
So my wife asked me this morning, is fog the same as being inside of a cloud? And I said, well, a little bit.
That's a great question.
I don't know the exact answer, but I'm sure some of our listeners know that answer. So, at any rate.
When you go through a cloud in an airplane, there's often turbulence.
This is true.
So things are happening in there.
Things are definitely
It's currently 15
happening.
Celsius.
Yeah.
It's cloudy.
It
and
is.
It's not getting much above 16 today here in New York City.
No, and it might drizzle a little bit, but tomorrow seems like a nice day, and Mother's Day on Sunday seems like a really nice day.
You know, I enjoy celebrating mothers of the world, but it is really a commercial holiday.
Yeah, I'm afraid it is. And Father's Day is like non-existent, except for lawnmower salesmen.
Say that again. What did you say?
No, because Mother's Day is a big deal, actually. People love to take their mothers out for dinner with the family and everything. And, of course, my wife will have a nice celebration, which I will participate in as well, for her kids. And but for Father's Day, which comes in June, that just doesn't happen somehow.
Nobody cares.
Well, that's not that they don't care. It's just that they do it differently.
Well, I ask on Father's Day to be allowed to sit down for an hour
by yourself
and do nothing
and veg out. No, I know exactly what you're talking about.
And I've tried this because by then the weather is good, so I sit outside.
That's a good time of the year.
And someone always comes, can you do
naturally,
this?
Naturally.
I don't mind.
Oh, well.
I don't need to be celebrated at all. I dislike actual celebrations of fathers when I'm the father or birthday. My birthday, I don't want to celebrate.
No, I hear you.
I just like to do virology.
and
parasitism.
Podcasts. That's right.
And this is This Week in Parasitism.
It is,
which is, you know, I'm sorry that we're recording a month after our last episode.
I know. We get into these ruts. I guess we're both busy people.
This time it's my fault because my course is wrapping up here, and I've done a bit of traveling, and your travel and mine were alternating, so we weren't around much.
That's true.
Now we should be able to be more.
This is very true.
We should get back to our twice-a-month pace, which we actually did maintain for TWiP.
True. That's right.
For a while. So TWiP is not dying, right?
No.
Neither are parasites.
Of course
not. In fact, yesterday on the train returning from Maryland, I found many parasite articles.
Did you?
Very interesting.
What were you perusing?
Well, I have a way of searching the literature. I get an RSS feed for each journal sent to an application.
Very nice.
Which I can then scan rapidly and save the articles I like.
Very nice.
It's a science, man.
It is science. So the American Journal of Tropical Medicine and Hygiene this week, which just came out, and I sent you an alert for that one, and I know you don't have an RSS for it, has a whole special section now on neglected parasitic infections of the United States. And it's interesting that the kinds of parasites that are identified as neglected, toxoplasmosis turns out to be one of them.
Is that what you sent me to mention on TWiP? Yeah.
Turns out to be another one, right? So, these are parasites that we've discussed in the past, but they're just not getting enough attention in the United States, I guess, because they're not perceived as being as health-threatening as some other things, which take precedence. At any rate, so that's why we're here, to bring attention to these neglected parasitic infections, because neglecting them ain't going to make them go away, as they would say. So that's
for sure.
They're not going away.
Dickson, the article you picked today was communicated to PNAS by William Campbell of Drew University.
This is true, and it was also produced by a good friend of mine who is no longer alive, Jerry Shadd.
University of Pennsylvania.
That's right. He worked at the vet school, and he was a wonderful guy and did a lot of basic work on the nervous system, of all things, of nematodes, of parasitic nematodes.
Dickson, is it nematode or nematode?
I say nematode, and you say nematode. That's fine. We could
Finding
write a
Nemo.
song like that, actually. There is a song like that for other things, but I say nematode.
Nematode.
So Jerry Shadd was while he was alive, was a world expert on hookworm. He owes his intellectual heritage back to the Hookworm Commission and people like Norman Stahl and those kinds of people
Which
that worked.
Talked about.
Yeah,
right?
we did.
Mm-hmm.
Yeah, we did. And in fact, I actually knew Norman Stahl. And that's basically how we came to know each other, because we attended the same sessions at the Tropical Medicine meeting and also at the American Society for Parasitology meeting.
I'm sorry he's no longer with us.
Yeah, me too.
Otherwise, we could podcast with him.
That's exactly right.
Are there any parasitologists at Rutgers?
There are. And I would have to actually look them up now, because it's a new generation of parasitologists there.
But we could go visit them in podcast.
That would be nice. That would be very nice.
I like visiting in podcasting.
Yeah, I understand that. You did something recently at an industrial site.
Yesterday I was at MedImmune.
Right.
Which is a biotechnology company in Maryland.
Right.
And I did a podcast with 4 of their scientists, which was pretty neat.
Terrific.
First time in a company for TWiP.
Wow. If we ever decide to go down to Washington, D.C., there's a lot of opportunities down there.
Yeah, we could spend a week just podcasting 3-4 times a day.
We could do that.
Put them all in the can and release them slowly.
There you go.
So this paper is a PNAS paper.
Yes, it is.
It was actually published a while ago, but that doesn't make it bad, right?
Of course not. No, it raises an issue that I wanted to address, and that is, the biggest issue is, how do parasites find their hosts? So there are multiple...
Maybe we find them.
Well, we do in most cases, all right? So let's talk about the routes of transmission just for a moment to review some basic information here. So this isn't different from bacterial infections or viral infections or fungal infections in the sense that you have to think about the route of transmission every time you think of an infectious disease, right?
Of course.
So like we have some...
Essential.
We have some bacteria that can actually penetrate the unbroken skin, don't we? Can you name them?
No.
Well,
I can't.
Syphilis is one of them.
Can penetrate unbroken skin?
This is correct.
It forms a chancre. Sexually transmitted disease.
And it forms a chancre at the site of infection.
Well, wait.
And that was not a broken portion of skin. That was just a mucous membrane.
Mucous membrane, though, that's different.
Well, that's still unbroken skin.
I mean, a mucous membrane is not protected. Like your skin, the outer layer is dead.
Yeah.
That's quite a barrier.
Yeah, no, that's true.
But, of course, if you cut it or scrape it or get a
bite...
Right,
but it's contiguous with the outer skin portion of humans.
What's contiguous?
The mucous membranes.
There's a dividing line. Anyway.
I mean, the structure is slightly
You're
different.
saying that Treponema pallidum can penetrate the...
unbroken skin, that's
Are you sure? Because we're going to get email.
No, I'm sure. Right. I am absolutely sure.
Fine. Viruses cannot penetrate the unbroken skin.
No, they have to be introduced by some other mechanism, right? Exactly. So you swallow them, or some vector bites you and
dog
introduces
bite,
them.
scrape.
Right, or they can be sexually transmitted.
Sure they can. But
those
mucous
abrasions,
membranes,
through abrasions.
The mucous membranes are ripe for invasion because they're a layer of cells just covered with mucus and a few other things. They're pretty vulnerable, But we have no choice. We need to be able to inspire and expire.
That's right. And we'll all expire someday without inspiring again.
The most common portal of entry for viruses is the respiratory tract.
Okay. So that's not true for most parasites. Most eukaryotic parasites don't infect that way.
Where do they like to do? The alimentary tract?
Yes. So a lot of them are foodborne. Okay. There's a plethora of parasitic infections.
Plethora or plethora? I agree with plethora.
I actually just learned what that meant.
Oh, come on, Dickson.
Do you know what that is?
Yeah, a lot of.
It's an ancient measure. It's an ancient actual area of measure that, like a cubit. A plethora is actually an ancient measure.
It's an excessive amount of something, not just an abundance.
No, I know that. That's how we use it now. But in the old days, it was used as an actual measurement of something. It was like, a ruler.
Really?
Yeah.
How much was it?
I don't know the exact amount, but I was watching some history show on television.
You watch
recently.
way too much TV.
I do. But this is late at night when no one else is looking.
Why don't you read?
I sometimes do.
TV is not going to help you.
You know what? I read all day long.
Yeah,
I sit at my computer
and I go home and read some more.
Well, I occasionally do that.
I dislike TV.
Well, that's
why I'm critical of you.
Well, that's okay.
Because I know I'm incorrectly applying my judgment.
Come on, I was No, let's not go there. Anyway, but okay. So the point is that the alimentary tract, the alimentary canal, is a major route of entry for eukaryotic parasites. So there's a bunch of them out there.
The parasite doesn't have to find us. It just gets ingested.
Yeah, that's exactly. Or drunk. Sometimes we drink them, right, in contaminated water.
It's accidental.
And there are lots of parasites that infect certain food items because that's their intermediate hosts.
Correct.
And then we eat that intermediate host.
Or sometimes we get fecally contaminated materials, right?
That's number one route. Number two route. The most common second route is vector-borne.
Yeah, that's interesting.
So you get reduvid bugs and tsetse flies and mosquitoes, and that's true for viruses too. It's a huge...
Are there any tick-borne eukaryotic parasites?
There are, yeah. Babesia is one of those.
Did we talk about it?
We did. It's related to malaria.
Okay.
We've talked about it a little bit.
I'm ashamed that I don't remember.
No, why should you? That's not important.
It's my life.
If you were missing a spleen, it would have been your life because without a spleen, Babesia is lethal.
Okay.
So that's
the vector-borne is number
joke
2.
I just thought I'd throw out there. So vector-borne parasitic infections are really very important throughout the world, okay, because that includes malaria, American and African trypanosomiasis. And don't forget the worms. There are some worms that are transmitted by vectors also. Onchocerca volvulus, we've talked about it. It causes blindness in its most acute form, transmitted by black flies, both in Africa and in Central and South America. We're eliminating it gradually, and maybe not so gradually. There are very few pockets left of autochthonous transmission for onchocerciasis. And it's possible that we'll eliminate it entirely one day because there's no reservoir host for it, right? Just humans. Same was for smallpox. That's the reason why we got rid of smallpox, because there was no reservoir host, okay? So those are two routes. But there's the filarial parasites, too, that are transmitted by mosquitoes of various kinds. So that's another big group of organisms that we have to discuss. And then there are the parasites that we find by accident, that we step on, let's say. Okay, there's one that we can step on that you can remember. It's called hookworm.
Hookworm.
Right. And the larva can actually, just like Treponema, it can penetrate the unbroken skin, but it does so by crawling down the shaft of a hair follicle.
Hair follicle, yeah.
Right. And so the related parasite to that is Strongyloides stercoralis.
We have talked about.
We have. But we made a distinction.
I'm going to find the episode.
Yeah, we can find that episode. And so we've made a distinction between hookworm larvae and Strongyloides larvae. And the big distinction is that hookworm larvae, for the most part, are not swimmers, okay? They're crawlers. So you remember the story I told about the finding out the conditions under which outhouses would function. If you didn't dig them deep enough, the worms could hatch at the bottom of the outhouse and crawl up the sides of the walls and exit and still become infectious for people that now went to that place to get rid of their infections.
Yeah.
And something else, of course. But then there's Strongyloides.
Strongyloides. Which we talked about in episode 23, Dickson.
23. The larva of which...
They call that a most unusual parasite.
It is a most unusual. It has two different phases of its life cycle. One can exist apart from a host in the soil, and the other needs a host.
Reservoir hosts include monkeys and dogs.
Monkeys and dogs. So one of the unusual features about the third stage larva of Strongyloides, which is not true for hookworm larvae, is this fact that it actually is equivalent to a heat-seeking missile.
This is a worm, right?
It is the worm. The worm actually has a well-developed nervous system, and that's where Jerry Shad actually did a lot of his pioneering work. Mm-hmm. He actually reconstructed in 3 dimensions the nervous system of the third stage larva of Strongyloides. And then he became interested in chemotaxis. How does this nervous system function? He became a nematode neurobiologist.
Where would you encounter Strongyloides?
You know, that's a very good question, Vince, because if you look around the world where Strongyloides is found, it's found basically everywhere. And in fact, it used to be a big problem for veterinarians that were doing maintenance of animal facilities for researchers. Monkeys that were captured and brought into the facility.
They were infected?
They were infected, in fact. And a lot of the vet workers became infected as the result of being exposed to monkey feces, which, of course, is semi-controllable, but, of course, not
always.
Not always, right? So they learned quickly that the very first thing you have to do is get rid of Strongyloides, and it took a long time for that to occur because the development of ivermectin was a recent advance, not an old advance. So to treat the monkeys was difficult, right? They had lots of other drugs to pick from, but the best one that we know of right now is ivermectin. And so Strongyloides is found primarily throughout the tropics and subtropics, but it also infects, in some cases, daycare centers for children.
Here in the U.S.?
Yeah, tropical, subtropical, temperate zone. That's correct.
Here in New York City, is there Strongyloides?
There probably is. And
if
the
we
reason
went to
why
Central
I say
Park,
that,
we could find it in the soil?
Well, I don't think it's going to exist too long there, especially because we have a winter. And a lot of these larvae died over the winter months. But they found 2% of the dogs of Montreal in one survey positive for Strongyloides.
That's a broad host range.
A very broad host range. That's right. And that's what we're going to discuss.
Are birds infected?
No. They're not.
A bunch of mammals?
Warm-blooded animals, that's right.
All right. So this gets in your hair follicle.
It does.
It goes to your lungs.
It does.
Then it comes up and you swallow it.
It goes through a migration route, very similar to the ascaris.
And it goes to your intestine.
And then it stays in your intestine. And remember, you only needed one worm in order to initiate this infection and get both male and female gametes produced, which then unite and form.
What's the downside of having this infection?
Oh, well, if for whatever reason you become immunosuppressed, this can trigger an explosion of the infection that's auto-infectious. It doesn't have to leave the body because apparently your immune system slows down the development of the worm to allow second-stage larvae to exit the body and then outside the body develop to the infectious stage, which is the third-stage larva.
Wow.
So the immune system doesn't function in a immunosuppressed state, and therefore the worms can develop from a second stage in the gut tract to a third stage in the gut tract, and now they're already home, so all they have to do is penetrate the gut tract and start this migration route again, and that's called auto-infectious.
But in a healthy host, you're okay.
No, healthy host, you're passing second stage larvae, and that's it.
And you do this for your life? Are you infected
Not for your life. No. Mostly your immune system can eliminate the adult worms eventually.
Okay.
So you self-cure, but in the meantime, of course, you suffer from perhaps some bacterial infections that the parasite actually drags into you on its way throughout your body. And
did you ever see this in your work?
Oh, yes, we did. And it was usually the tragic cases that we got to see because they were in the hospital for another reason. They were from a tropical or subtropical country. One in particular, I remember, came to the dermatology service, and she had this enormous rash across her abdomen. And when they biopsied it and looked under the microscope at the slides, all they could see was third stage migrating larvae. They were everywhere. And they didn't actually know right away what they were looking at, so they, raised their hand, and I came over and helped them look at it. And sure enough, they were Strongyloides larvae. And this woman had received an immunosuppressive therapy for some other reason, without ever checking to see whether or not she had Strongyloides. Other patients had come in for transplants, right? Like kidney transplants or even cardiac transplants from areas where this infection is endemic. You'd have to check them out for Strongyloides before you even started the immunosuppressive therapy to get them prepped to have the organ transplanted. So there's a rich literature on this in the early 60s and 70s and through the 80s as to how to warn surgery groups to the fact that your patient might die not from a rejection of a heart, but rather from an overwhelming bacterial infection of some sort, initiated by these migrating larvae during an auto-infectious cycle. Because when they're in your large intestine, they're eating until they get to be a third stage larva. That's how they get to be the third stage. And what is their gut tract filled with? Your microbiome. And what do they do when they go into the bloodstream? They transform to the third stage, and in doing so, they empty their gut tracts. So wherever they are, when they develop from a second stage to a third stage larva, off comes the cuticle, out comes the new larvae, and all the bacteria in their gut tract is deposited wherever they are.
Wow.
So they're you're looking at them as a micro syringe filled with mixed microbiome. And the brain is a horrible place for this to happen because there's almost no immune reaction.
Now, their microbiome would depend on where they've been before, right?
Well, everybody has a unique microbiome.
Has anyone studied the Strongyloides microbiome?
No. I don't think Strongyloides per se has a microbiome. I think
you might.
Well,
I bet it does.
Cenoabitis has a microbiome. I've learned this recently.
Some insects have a microbiome consisting of one species.
But that's remarkable.
And those are nice to study. It's easier to study.
Oh, yeah, sure. So would you say that Onchocerca has a microbiome because they have Wolbachia?
Why not?
Isn't interesting? Sure. But
I mean, it's a symbiont, right?
So the gut tracts of parasitic nematodes.
Yes.
As far as my mind can think back and say, gee, I've never encountered a bacterial species inside the gut tract of. And I can prove to you that Trichinella doesn't have a microbiome because I worked with germ-free animals and I could sterilize them just by allowing them to sediment through successive changes of sterile saline before I actually put them inside the isolator. You could wash away the bacteria from the worms, and then you can infect germ-free animals, and the only thing they would catch would be the Trichinella infection rather than Trichinella plus their microbiome, because they don't have a microbiome. Okay, so I don't think hookworm adults have microbiomes, even though they live in the gut tract. They clump off pieces of tissue. The Strongyloides parasite at an adult stage lives embedded in the columnar cells of the small intestine. Again, I don't think it has a microbiome. But the second stage larvae have to grow up to be third stage, and that's the feeding stage for this worm outside the body, okay? Normally, they would feed on feces outside, transform to third stage larvae, and wait for the host. But here's the difference.
Yeah.
Now we're going to get to this article because hookworm larvae will quest. Remember we discussed these aggregations of third stage larvae, and you get these large clumps of them crawling up to the highest point in their local environment and forming these massive writhing I'm trying to paint a picture here for our listeners of these. It almost looks like a flame, a slow burning flame. But when you look close, these are hundreds to sometimes thousands of third stage hookworm larvae that are sitting on the tops of blades of grass for the most part, waiting for someone to come along. Strongyloides is an impatient parasite. It doesn't wait. Strongyloides, instead of questing, swims. It's a swimmer, a long-distance swimmer. So if it's a swimmer,
then swimming?
It swims in water.
Really?
Fresh water. So let's take the tropics, for example.
Like this?
Yeah, sure. No, it's a serpiginous swimming.
On the surface or deep down below?
Well, that's a good question. No, neither one. It follows a trail. of chemicals.
Nah, I don't believe it.
Well, you're going to have to believe it because that's what this article is all about.
Wait. It's in the water.
That's right.
There's chemicals everywhere.
So who else?
So
is
maybe
in the
there was
water?
a mammalian host swimming, and it's going to follow.
That's right.
Yeah, but the host is much faster.
It doesn't even have to be that much water, by the way, because no, these are little kids that are sitting in puddles of water, basically outside in the mud.
Oh, and these worms are swimming towards the kids.
That's right, because their environment's contaminated with human feces, and there's monkeys in their environment sometimes, and dogs. I mean, when I visited India, for instance, many times, the monkeys are everywhere. There's a subset of primates
yeah, of course,
that we're supporting, and they're all over the place. They're on the roofs, they're in the lawns, they're everywhere I went in India. I saw monkeys everywhere. So monkeys can carry Strongyloides.
Sure,
right?
sure.
And it rains in India. Of course, they have monsoons, and they have seasons for this. They have dry seasons and wet seasons. But can you imagine little kids, hey, it stopped raining, let's go out and play, because they haven't been out of their house in a while because it's been raining like cats and dogs. So they go out, and they start playing, and they get wet. And, of course, they defecate outside, and the animals defecate outside. And you can easily see how these parasites can survive in those situations quite nicely. In fact, they depend on those situations. So a little kid sitting in a mud puddle that's been contaminated with human feces, that has Strongyloides larvae in it. Maybe they have, you can get this now because they have a life cycle in the soil which allows for the multiplication of this parasite like crazy without a host. And then when they reach a certain level, when they run out of food, they all arrest in the third stage larva. They just say, I don't want to go any further because I don't have any more food. I'm going to wait for it to rain. And when it rains, I'm going to swim. And I'm going to swim around until I detect the host. Now, this article that we're about to talk about says, how does it detect the host? What part of the host is being detected by parasites?
Okay.
So,
and the model system is
a dog. Because dogs...
I'm not going to read the title because it'll give it away.
I know.
Let's not read the title.
No, we're not going to read this title.
But we'll say
later.
who the authors are. Daniel Safer.
Right.
Mario Branis.
Right.
Seth Dunnipace.
Yep.
And Gerhard Schad.
Yeah.
Your friend.
Yeah. Jerry Shad.
Jerry Shad?
Jerry Shad.
You were his good friend? Or?
I was a very good friend. Yeah. No, I've even stayed with he and his wife many times.
Yeah, so the model is the dog. And the question is, how does a stercoralis find the dog?
Or a monkey or a human. So we have to have something in common with those three hosts in order for this parasite to be so broadly based in terms of its host range. Okay? So what did they do? They realized that for the most part, the logical sequence of thinking would be, it's got to be something in the skin of this animal, right?
Wow, that would make a lot of sense,
It
wouldn't it,
would, but
Dickson?
there's a parasitic catfish, for instance, that everybody has heard about, I guess, that's heard about this from South America, and it's called Kanduru.
That swims into your penis.
Yeah, well, or into your vagina, depending on which sex you are. It's not following a trail of what this parasite follows. Mm-hmm. It's following urea.
Urine, yeah.
No, not urine.
Well, urea is part of urine.
Well, it's a chemical in urine.
Yeah.
So it follows a trail of basically ammonia because the urea actually breaks down in the environment. And so these parasitic catfish, they're looking for a gigantic catfish. And what they're looking for in the gigantic catfish is the gills. And the gills are the excretory organ for volatile organics.
Including urea.
Including
How
urea.
big are these guys? What's the name again?
Kanduru.
Yeah, we've talked about this before.
Yeah, we did. They're tiny little guys, right? With barbs sticking out from their pectoral fins and their dorsal fin, which allows them to go into the gill rakers of these large catfish, and then they just stay there.
Toothpick fish.
The toothpick fish, right?
We actually Candiru.
Yeah, the Kanduru. We had a case of that here.
Really?
Yes, we did.
Oh, it looks like a toothpick. It's very long.
It's very long. It can swim up, but it can't swim back.
So it swims into your urethra.
That's right. And then it gets stuck because it can't reverse itself.
And meanwhile, you're screaming.
Quite loudly, I'm sure.
Wow.
I know. So that's one chemical trail that's laid down by the host that this parasite, in this case, a parasitic vertebrate, follows in order to capture its host. Okay, so then we discussed another parasite which swims called schistosoma. So that's a trematode. Okay, it's not a nematode, it's a trematode. Right. But it contains a stage which is produced by an aquatic snail called the Cercaria. And the Cercaria is a swimming stage. And in this case, the Cercaria, which hatches from the snails in slow-moving water or lakes throughout the tropical world, mostly through Africa and South America, but also in Southeast Asia for the other species of schistosomes. This stage is responsible for completing the life cycle. And so it swims to the surface of the water first and then tries to detect lipids, which evolve off of the surface of your body as you enter the water.
Makes sense. Yeah.
As you enter the water, the lipid layer of your body extends out in all directions from you. Right. And you don't think about this.
It's an oil slick, right?
It's an oil slick. We create an oil slick every time we step in the water. And if we stay still, these Cercariae that are swimming randomly throughout the water column, not the water column, I'm sorry, throughout the surface of the water, immediately detects the molecule. In this case, I think it's lanolin. And lanolin is one of the components of skin, right? So it starts to zigzag its way, and that's how they find out where the highest concentration is, because they're busy. No, it's not there. Oh, it's there. It's more. And it's sort of like a heat-seeking missile. You know, it sort of homes in on the signal, and then it just heads right for that signal.
It's pretty cool.
So that's very cool.
Yeah,
that's
like that.
Extremely cool, right? And you can watch them do this. If you make a slide, a microscope slide, and you have lanolin, and you make a preparation of that, and you smear it so that you create a gradient, you can put the Cercaria at one end of this thing, and you can actually watch them move towards the highest concentration gradient.
What was the parasite? This was the
schistosome.
Schistosome.
Yes. Now, there have been some controversies over the actual substance in the skin that attracts the Cercaria to the skin. So this data may have been upgraded since I just told you that lanolin was one of those substances, but it's certainly one of the lipids that we secrete in our skin. We have a lot of sebaceous glands in our skin for our own protection, basically. It makes our skin pliable and waterproof. And those are two interesting characteristics of our skin that we treasure and value.
So there we are, two chemoattractants, urea
Correct. Right.
and lanolin.
That's right.
Now, two different parasites. So now we're going to look for the Strongyloides attractant.
Yes, we are. So they start naturally with skin.
With a dog.
With a dog, because the dogs are easier to manage in the laboratory, so they begin by extracting dog skin.
Now, wait a minute, Dickson. Do they have to kill the dog to do this?
I hope not.
Canine skin from laboratory animals euthanized for other investigations.
Okay.
So they
didn't kill the dog, but somebody else did.
Somebody else did. So you can imagine you can get a lot of skin.
Sure.
And they made an extract.
They did.
They call it an aqueous extract.
Yeah, because it has to be aqueous. Otherwise, if it's a lipid extract, right? If they don't use a bipolar reagent to extract the skin, then it probably isn't relevant to the way the parasite detects the host, because it's an aquatic environment that the host is in.
Okay.
All right. So they have to start with water-soluble or at least water-extractable elements of skin.
Course.
It makes sense.
So they extract it.
They did. And they have a crude extract of dog skin. And then they test it in some migratory system.
And
right?
What would that be, Dickson?
Well, you have a lot of choices here. You know, I actually did one of those myself. I did. Again, this is a topic that we've touched upon in many of the episodes, but it's good that we can bring this together here, because there's another component of the environment which parasites can detect, and that's heat. Believe it or not.
Yeah, of course.
Heat. So thermotaxis is
carbon
a big
dioxide
deal.
too, no?
Well, we'll talk about that later. I think
in another session.
It's probably tougher for a nematode to detect CO2 than it is for them.
Mosquitoes do, right?
Mosquitoes definitely do. There's no question about that. So now we've got heat-seeking missiles, and that's definitely the case for nematodes. All parasitic nematodes that have been tested for this characteristic have been found to be positively thermotactic.
Right.
And the free living equivalent nematode group, the The Xenoibriduses and the Neoaplectanoglazari, which is a parasite of cold-blooded insects, invertebrates, I should say. They do not have a positive thermotactic response. And you can set this up in such a way as you have a saline water bath, right? Mm-hmm. And you heat one end and you keep the other at room temperature. And what you do is you put the sample of worms in the middle, okay? And then you have to have an observation system. You can watch them migrate.
Yeah.
And free-living worms will migrate to the cooler end, and the parasitic worms will migrate to the hot end. The remarkable thing about this, Vincent, is that I'm going to, with my hands now, show you how long this migration chamber is. It's about a 1.5 to 2 feet. If you put a tiny little parasite in the middle of this gradient, they can always hit the hot end of this thing. Always. Do you imagine what the gradient actually looks like for temperature? What if I had a 10-degree temperature difference? If you measure the gradient per millimeter or millimeter is about the best way to do this. The temperature is so small. The differences are so small. This parasite must have an exquisite temperature sensing device to detect which direction to go in. So their nervous system is really fine-tuned.
Yeah.
And so adult Trichinella, that's the way I used to collect adult Trichinella.
How would you do that?
I put them in a circular dish.
Yeah.
And I insulated the bottom except for the middle. And then I had a wire mesh insert, which was hollowed out in the middle also. And I took the gut tracts of infected rats. I opened them up and I washed them very quickly in saline to get all the debris out because I kept them off of food the night before to make sure that their gut tracts weren't filled with food. But they were infected with Trichinella. And I would lay them on the wire mesh on a heating pad. And so this asbestos insulator in the middle would only allow heat to emanate from the middle and go out in all directions. And this was, I call it the circular thermal migrator device. And it actually worked brilliantly. It was fantastic to watch it happen because I would have like 20 or 30 gut tracts of infected rats in this migrator. And in 20 minutes, you could see streams of adult worms that had fallen down on the bottom.
20 minutes.
Say, 20 minutes. It was easy to watch. And I've got some photographs of these times 0 and times 20 and times 30. You can see a big ball of worms in the middle eventually.
Cool.
And they didn't get there by convection current. They got there by actually detecting the gradient and migrating towards the hot end.
Nice.
So that's how you did that. So, yeah. So a guy by the name of Ralph Thorson and his graduate student, they actually discovered thermotaxis in parasitic nematodes.
All right, so now we have another attractant. Gradients.
Correct. So their nervous systems are attuned to their environment and allows them to succeed as parasites by keeping track of the only environmental influences that will result in an infection. Okay? So now we've got this swimming nematode. Now, swimming trematodes detect other things. So what does this one detect? And they said, as long as we've got this family of parasites, and all of which respond to their environment to ensure success of the transmission cycle, why do I wonder what Strongyloides detects?
Okay.
Right?
So we make an extract of dog skin.
We do, and then we set up a migration chamber. So the migration chamber is actually a very low agar-containing...
It's a Petri dish, right?
It's a Petri dish, but it's very low concentration of agar to allow maximum rate of diffusion of factors. So you had a central well that you create with a little cutter, and then a well on either side of that, equidistant from that well. You put your substance that you're testing in the middle well, and then you put worms on either side. And so you've got two sets of results that you can measure, right? And eventually what you're going to look for are worms that you put in the outer wells, and you ask whether they randomly move about in the well or in the agar, or do they actually migrate towards the source of something that you've put in the central well. And that's the migration chamber that they use.
And they can take photographs of the trails of
They can do that. Well, they had to do something else for that one. They had to put a grid over it with a black background, and then they measured the rate of migration. And they could actually if they had, they could have taken a video.
Where do you get the Strongyloides from? You buy it?
You don't have to buy Strongyloides.
you
can raise them in the lab.
You can?
Because it has a free living cycle, remember? You don't
culture,
need a host.
In culture medium.
You don't. What they used in this case was dog feces and charcoal. A 1-to-1 mixture, dog feces and charcoal. And the dog feces contained, those were infected dogs, of course, with Strongyloides. And then they would allow the life cycle to go on inside the Petri dish. Mm-hmm. You know what you have to be careful of there, Vincent?
No.
This is a swimming parasite that can actually crawl out of the Petri dish. Yeah.
Really?
Yeah, so investigators found that out pretty early also.
They got infected?
They did.
Can you see them with the naked eye?
No, these are very small.
Yuck.
I know, no kidding, right? You have a Petri dish with a cover on it, and you've got a charcoal fecal culture inside the Petri dish, and you say, well, it's pretty safe. I guess I can work with this, but you should wear gloves always, no matter what. The people who didn't wear gloves right away, they can crawl right over the lip of the Petri dish.
How would they know they were infected? Months later or days later?
Oh, no, they would know earlier than that because this causes a diarrheal disease.
And they would go, uh-oh.
Exactly.
Oh, dear.
Exactly right. So that's the migration chamber that they set up.
Yep.
And lo and behold, the very first time they did an extract of dog skin, they got a positive result. The larvae actually migrated, third stage larvae. Second stage won't do this because they're not the infectious stage. Third stage larvae will migrate towards the gradient created by the concentration in the central well. So that's cool. You know
what else they did that was cool?
Go ahead.
They had an extract of gerbil skin.
Okay.
And that worked.
Okay.
Because they're known to be hosts.
Okay.
They had an extract of cat skin.
Right.
Which are not hosts.
And
what do you think happened?
My guess, and since I reviewed this paper for PNS, I guess I think it didn't work.
Did you review it?
I didn't review the paper, but I was asked
You made a commentary.
Yeah, I was asked to write the commentary on it.
Is that the last thing you ever wrote, Dickson? Academically?
Hopefully not. No, of course not.
It's called Chemical Trails and the Parasites That Follow Them.
That's right. Or another way to put it, and I really wanted it to be that title, but I didn't have the nerve to
Tell me, we'll use it for this TWiP.
It's called Happy Trails to You.
Should we use that as a title?
Absolutely.
Happy Trails to who? To Strongyloides?
Happy Trails to you. You're leaving happy trails for the parasites to find you.
Oh, that's very subtle, Dickson.
Well, I'm a subtle kind of guy.
So they have a attractant assay.
Yep.
And what do they do next?
Well, in the positive ones, of course, they would like to know the specific ingredient or ingredients that are actually responsible for the parasite's ability to detect them. So they begin to fractionate the skin. And there are lots of components in skin.
The extract, you mean?
Yeah. There's a ton of them. But there is a dominant chemical in skin which they detected as a positive response in Strongyloides' case. And, of course, you'd expect this. I mean, it's almost self-fulfilling. What would parasites home in on or be selected for? But the most common shed ingredient to your skin would be probably the one that you'd expect evolution to favor, because the minor ones that are not shed as plentifully or perhaps at all would not be the ones that you'd expect to be the ones responsible for this. And in fact, they found out, should we now tell them the name of the compound?
Well, what I find interesting, before you do that,
they
actually used chromatography of various sorts to purify this. And then they had so they would fractionate.
So Jerry didn't do that part of the work. These other guys did that part of the work.
They would take the fractions and test them in this migration assay.
Right.
And eventually they got a pretty pure fraction, which they then submitted to mass spectrometry to identify it.
Yeah, that's right.
Gas chromatography, mass spectroscopy.
It's become so easy.
138 kilodaltons.
It's amazing.
And then that's easy to identify it. And what is it, Dickson?
It turned out to be urocanic acid.
Urocanic acid. Now,
correct.
As far
so
as
I went and looked up urocanic acid because I didn't know what it was.
Looks to me like uro is urine and cano looks like dog, doesn't it?
Yeah, but it's not that. That's not where that name derives from, and nor can I tell you where.
That's where it comes from.
You think?
I know. I looked it up.
You did?
Yeah.
Well, because it's a normal component of human skin, too, though.
But it was originally found in the urine of a dog in the 1800s.
I see.
That's why it's called urocanic acid.
Take it all back.
Canis is dog, right?
Take it all back.
Anyway, what were you going to say?
So
it's a major component of skin.
What was the very first organic chemical synthesized?
Urea?
That's right.
Is that the Haber reaction?
No. No, it's not the Haber reaction. That's nitrogen fixation.
Oh, yeah. What's the urea reaction? What's the
No, urea was the first organic chemical ever synthesized.
What's the guy's name?
Voller.
Voller synthesis, yeah.
Yeah.
We talked about that on urban agriculture.
Yeah, we did that. Okay, but by the way, this parasite interfaces very nicely with the agricultural interface as well because of rice growing in Southeast Asia.
People get infected, right?
This is a swimming parasite, Vincent. Yeah. If you have to go to the bathroom, where are you going to go? And you're stuck out in the middle of a rice field, and it's only 12 o'clock, and it's not even time for lunch yet, I think you're going to defecate in the rice field. And you know what?
Oh, my gosh, Dickson.
The rice grows a little bit better because you've just fertilized the rice field. It is gross, but that's the way it is. That's the way it is.
That's life.
That's life in the raw.
So you know what they found in this paper? They found that metal ions inhibit. So they eventually got purified urocanic acid, and they showed that it attracts, and that metal ion inhibits the chemoattraction.
Huh.
How do you think that worked, Dickson?
I don't know, but you'll tell me.
It binds to the urocanic acid, and apparently masks the ability of the Strongyloides. Stercoralis.
SS. We used to call it SS.
SS.
Yeah.
To recognize it.
That's right.
Now,
wait.
Okay, I'll wait.
We owe the listeners at least the courtesy of telling them what we think urocanic acid does for us.
Yeah, that's a good question.
What does it do? What is its function? Why are we so blessed with so much urocanic acid in our skin?
Well, I can take a guess.
Try.
I'll bet it prevents UV rays from burning us or doing.
You know what? You've looked
cancer.
This up, haven't you?
No,
you've done your homework.
Would you expect otherwise?
No, I would not expect otherwise. Of course. And it's a histidine derivative.
Yeah, it is.
And it's enzymatically produced from a larger molecule which contains polyhistidine.
Is it formulated into sunscreens?
You know, that's a great question because it's a natural sunscreen, obviously. What it does, it prevents UVB radiation from creating thymine dimers.
Yeah.
Which is the major mutational event which results in melanoma formation. All right, so that we think, okay, therefore we are, we think that urocanic acid is a big player in the evolution of humans.
Yeah.
Well, guess what?
But dogs have it too.
No, but the other thing is if our parasites can home in on that.
Well, yes.
How do we out-evolve them? We can't.
Yeah, because we can't get rid of it.
Well, we can't certainly.
Unless we live in a cave.
We can. That's right. We're troglodytes.
And then we wouldn't worry about sun. Eventually, we would evolve not to have urocanic acid anymore because there is an energy cost to making it.
There is. So that's an interesting second look at the same molecule. Okay, so urocanic acid turns out to be the chemoattractant for Strongyloides stercoralis.
Do you think it's the only one? Do you think there are others? How would you do that experiment?
Well, you'd do
I'll tell you how I would do it.
Oh, well, go ahead. Tell us how you would do it.
But
then.
You can tell us how you would do it.
I would make a preparation from skin, and then I would eliminate urocanic acid, but nothing else.
Yeah,
and then
that's
I would
a good
try
one.
That one.
That's a good one. My approach would be to identify the receptor for urocanic acid in the Strongyloides and somehow block that or delete it. More complicated than your approach.
You could block it, all right. I mean, you could probably block it by flooding it with the smaller urocanic acid molecule.
That's one of the things I looked for. I searched Google. I searched PubMed, and there's no other papers on Strongyloides and urocanic acid since then. So no one's looked at the receptor. But that would not be so difficult to pick up, you know.
Not so difficult, but at the same time, I mean, I think you would probably have to use a confocal microscopy and a radio-labeled or a fluorescent-labeled urocanic acid.
I would label urocanic acid and try and pull out the receptor from extracts of Strongyloides. Well, want to do it? We'll write a grant.
Sure. To get enough Strongyloides to do this experiment would be challenging, to say the least.
Yeah, that's what the reviewers would say. He has not provided evidence that he can
safely culture Strongyloides without infecting the entire floor's worth of people.
So you'd have to have those experiments done ahead of time in order to get this funded.
Or maybe you could do this in Caenorhabditis elegans if you could get the genes for the receptor transferred into Caenorhabditis.
Yeah, but to do that, you need to have
now you can get a lot of
Strongyloides to begin with to get the receptor.
Well, they have the genome of a lot of nematodes, and I would be surprised if they wouldn't work on Strongyloides' genome because of its unusual characteristics.
So, Dickson, what's the utility of this observation? Can you use it to prevent infections?
It's got potential for that.
Is anyone working on preventing Strongyloides infections? No, it's a neglected disease, right?
It's a neglected disease, but the way you prevent it is through good sanitation. But even with good sanitation, with all these reservoir hosts, you're going to have a problem. And because it swims, you have another problem. So here's an episode.
I know how you get rid of the water.
Yeah, right. So there was this. In fact, I think in our episode on Strongyloides, we discussed the invasion of Grenada by the United States Marine Corps. And the movie that was made of it was starring Clint Eastwood.
Mm-hmm.
And what they found, forget the movie, but what they found in the 6,000 returning Marines that were involved in that operation was that many of them in Grenada had acquired strongyloidiasis.
Wow.
And they remember jumping into foxholes that were created by the Cubans that were being kicked out of Grenada by the U.S. Marine Corps. They found their foxholes, but it had rained, and there were watery environments in the foxholes. Now, I'm going to tell you something here, Vincent, and you're not going to like this at all, because if you didn't like the farmers and the rice paddies defecating to fertilize their fields,
the guys defecated
it's
in
even
the foxholes.
Worse. If someone shot at you and you were in a foxhole,
you'd take a dump, right?
And you had to go to the bathroom. You said, "Hey, time out. I'm sorry, I have to go to the bathroom. I'll be right back." You can't do that in a war zone.
No, I understand.
So you were going to relieve yourself at the site of
it's war. War is hell, Dickson.
War is hell. And in this case, the Cubans did it and the Americans did it. And the mixture of materials in these foxholes was biological.
Sure.
So a lot of them came back, and they all had to get string tested. And I think it was at Fort Bragg for this parasite, for Strongyloides, and they were all treated. And I hope that they all resolved their infections, basically. But remember, in war, what is another thing that happens? Your adrenal glands go crazy, and that's immunosuppressive. So you're now susceptible to some diseases that would ordinarily be infections, but now they become
have
diseases.
A solution.
So you become hyperinfected sometimes if it's a protracted war. And you could die, not from a bullet, but from a nematode infection.
I have a solution.
You have a solution. Stop fighting.
Yeah, how'd you guess?
I can see it in your eyes. I was going to give you the same solution. That's about the hardest thing for human beings to do, unfortunately.
Dickson, do you think this would be a chemoattractant for any other parasites?
There are very few other nematode infections of humans that occupy, that engage in this particular transmission scenario. So I think the answer is no. I think this is a limited engagement, as it were. It's not even.
All right.
It's not even from hookworms. All right. This is just Strongyloides. All right.
Okay, but it would be kind of neat to check out. No.
No, it sure would be
easy enough to do.
No, absolutely right.
Is that the story?
That's
basically
the story. So the reason why I wanted to resurrect this article, because it is an older article, is that it occurred to me that we haven't discussed the general principle of how parasites find their hosts. We've talked about niche selection, and we've talked about immune responses against them and stuff like this. But we never really broadened the scope of the discussion to include how do parasites find their hosts. And one of them, of course, we said delightful, because they find each other in the tall grass.
They find each other delightful.
That's right. That's how they find each other. But this is how parasites find their hosts.
I think that's a really good topic that we have ignored, and this was a nice little intro.
So we did it. So I feel happier.
Very
But
good.
I'm sure we're going to get a lot of interesting stories from our listeners about their own experiences with host-seeking activities from parasites.
Absolutely. Would you like to read some email?
I would love to, but my iPad does not allow me to. But if you'll turn yours towards me, I would be happy to help you read
Well,
these.
I don't want to turn it.
No, okay. You can read them. I'll answer the questions and you can read them.
I might turn it.
All right.
All right, the first is from Heather, who writes, "Hi, Doctors R and D.
R&D, I like that.
I thought you might be interested in this news article about aquaculture in Hong Kong. Perhaps it's time for another fish parasite episode. I love the podcast. Keep up the great work." So this is an article from BBC News, Hong Kong's fish farms in the sky. Apparently they have fish farms 15 stories up in skyscrapers.
Okay.
11 tanks, 80,000 liters of salt water. They're
That's
growing
vertical farming.
grouper.
That's vertical farming.
Yeah. A
Go
vertical
on.
fish farm, it's called. They call it that here in the article.
How
I think
about
you should
that?
sue them because you
No,
have...
no. I want them to use the words. I don't want to sue them. But what do I do that for?
Now, the interesting thing here is that, anyway, we'll do some fish parasites, right?
Sure. We already did. We did some already.
Yeah, no, that's why she said we should revisit them.
Right.
Now, here is, it's not just fish farms that have been taken to the skies. As a number of organic fruit and vegetable plots are being created on top of skyscrapers.
Okay.
And look, they have a photo here.
How about that?
Isn't that cool?
Yeah, no, that's very
Rooftop
cool.
gardens.
It's very cool.
Is that urban agriculture,
You bet it
Dickson?
is. You bet it is.
Well, if you go to urbanag.ws, you can check out our
This is
podcast
very
on that
true.
topic.
Yeah, on that topic. That's right. So here's a crossover topic. That's
And
great.
look what else they have on these rooftops. Do you know what that is?
Looks like bees.
Bees.
Sure.
Put bees up there.
You
Why
get
not?
bees in Brooklyn also.
I guess space is at a premium.
Sure.
The urban farms reveal how shallow Hong Kong's urban roots are.
That's interesting.
So urban farms,
Yeah.
rooftop gardens, beekeepers.
How about that?
This guy who has a company, HK Honey, who
Nice.
puts
Nice.
them up on the roof.
I guess HK stands for Hong Kong.
It does. And
Sounds good.
So that's pretty cool. This is actually relevant to urban agriculture.
Right.
Vertical fish farm. All right, now this one you should read because
All right.
it's about trichinella.
Oh, dear.
I'll turn my laptop around for you.
All right. Would you
Oh,
please?
look, the cord isn't long
No,
enough.
it's long enough.
Can you read? Is that too far away, Dixon?
I can get closer.
Yeah, but don't go away from the mic, okay?
It's Bill writes, Trichinella, 1835 to 2007. After a long, productive life, I am sad to tell you poor Ella is gone. It seems she was always around, and that she had such an infectious nature that she would never fade into history in this manner. We need an update. Your web guy can set an auto updater for dates and copyright notices, etc. So as each year passes, the year is incremented upwards. Each date you update needs to be told to the web guy so it is included in the update and does not change the font or color. It makes the website look fresh. I must say that I haven't updated the website in a while because...
This is trichinella.org, right?
Yeah, it's trichinella.org.
So he's complaining of the fact that the most recent date you have there is 2007.
No, that's true because the data that was sent to me for very nice. Ray Burristan in Florida used to send me CDs with the yearly updated literature from all of the articles produced on trichinella. And at one point they became non-translatable to an easy RSS upload, and that's the reason why it stopped at 2007.
We can fix that, Dixon.
We're gonna fix that.
I'm going to take over all your websites because I don't have enough to do.
Yeah, exactly right. So no, I leave it up there because it's still instructional with regards.
Well, there's
to
a lot of
life
stuff
cycle
there.
and
Yeah, it's great.
Epidemiology is still valid, and there's a lot of valid stuff on it. But I
Look,
appreciate
we
it.
don't need to update the literature because people can go to PubMed, but there are other things there that are valuable that we will take care of.
Well, that's the complaint, is that we haven't updated the
Yeah,
literature.
well, there's a date there on the front page. We just have to take that off.
Right.
Anything else from him? Is that the end of his letter?
That's the end of his letter.
All right. I'm going to take this back here and read the next one. You need
Right.
to really fix your iPad.
I need to remember my password.
Ellen writes, oh, this is very cool. This actually came a while ago, and I used it for TWiP because it is a cross-stitch. It's a woman who does cross-stitch
Yeah.
Alicia's Etsy shop. She's Ellen writes, anyway, thought you'd enjoy this. It's the malaria life cycle in a cross-stitch. Here you go, Dickson.
Oh, yeah.
Do you know what cross-stitch is?
Yeah.
I don't think you have any clue, do you?
It's a way of embroidery, I think.
Anyway, look, the whole malaria life cycle.
It's wonderful. No, it's great. Love it.
I wonder if she has Strongyloides, huh? I could use that as the
I bet you she might, but it's a more complicated cross-stitch, that's for sure.
Cross-stitch pattern of basic life cycle of malaria with mosquito and 4 forms.
Nice.
So basically, it's a pattern
that
you can buy, and you can
cross-stitch
it yourself. It's only $7.99.
That's not bad.
Now, so we want to thank Ellen for sending this. She writes, Alicia Watkins' Etsy shop has many cross-stitched bacteria and viruses too,
which you're familiar with.
Yeah.
Right.
All right. Let me turn my laptop around and let you read the next one.
All right.
Are you able to do that, Dickson?
Sure. Fish pathologist writes, Dear Doctors R&D, I'm catching up on old episodes. I very much enjoyed Dr. D's reading from his new book. I see that his Vertical Farm book is in audio format. Will the new book also be available in audio format? I don't know. Dr. D telling stories is one of my favorite parts of TWiP, and I would love a whole book of them. Don't tempt me. I also wanted to tell you how far-reaching your podcasts are. My husband works coordinating volunteers for a state park. A science club from a community college in Tennessee came to volunteer in the park during their spring break. My husband was very taken with how industrious and intelligent the group was. I worked managing chemistry and microbiology labs for undergraduate at a state college, so I was naturally slightly suspicious of these assertions. The students and their advisor invited us to have dinner with them, and I must admit I found them very engaging young people. Due to my interest in previous work with parasites and microbiology, the conversation naturally turned in that direction. One of the young ladies, who couldn't have been more than 18 years old, mentioned that she absolutely loved all the TWiP, TWiV, and TWIM podcasts and is pursuing microbiology as a field with an interest in cyanobacteria. I thought you would enjoy hearing how your work inspires some of the youngest of the new generation of scientists. Thank you for the excellent podcast. I look forward to more stories, and I really like having guests like Sagi in TWiP.
We'll have some guests. Actually, Sagi's old boss at the UPenn, I wrote him.
Yes.
Because he heard Sagi on, and he said, I'll come on whenever you want. So I said, okay, come on. He said, actually, we have a young faculty member who you should have on, who works on malaria.
Come on, we can do this.
We'll do that.
Sure.
I think this is great that these young kids are listening to us.
That's right.
I love it.
Yep.
And it's not just about me, Dickson. It's about them.
That's true.
Fish pathologist. Ellen writes, Hi, Vincent and Dickson. I enjoy the TWiP podcast as someone who's interested in science, so please excuse me if bubonic plague isn't a parasite.
Ooh, I wouldn't say that.
It's a bacteria. It's not a eukaryotic parasite.
Well, it's not a eukaryotic
but
parasite.
it is a bacterium.
It is a bacterium. And it
but if it is, did you see the article in The Guardian where archaeologists and forensic scientists say only an airborne infection could have spread so fast and killed so quickly? What do you think? Does this make more sense than transmission by rat fleas?
Well, I think rat fleas start the process, and then it becomes from bubonic pneumonic, and pneumonic plague is airborne, and that's what starts the pandemic in humans. That's what does it. That's how it works.
This article is called Black Death Skeletons Reveal Pitiful Life of 14th Century Londoners.
Yeah.
DNA from emaciated London Black Death skeleton matches modern plague bacteria and supports airborne theory of spread. So that basically there's nothing different about the strain that was there back then.
But they had no antibiotics, so therefore they had to suffer. Today, penicillin will cure that infection.
Today, Dickson, can you still get plague?
Oh, yeah.
From whom?
You can catch it as the result of being bitten by Nosophila fleas that hopped off the body of a dead prairie dog or a squirrel out west. And there have been lots of cases of plague reported to the Centers for Disease Control.
So you get bitten by a flea, and then can you transmit it to someone by respiratory route?
No, not right away. The bacteria lodges in your lymph nodes, and then they start to necrose, and that's where the buboes come from. That's what they're called. And then eventually, if you allow this to go longer, then it could become pneumonic, because the buboes will then spread the infections to your lungs. And if you breathe on somebody else, you can transmit it via the droplet infection route. So I remember my microbiology.
That's why I put it in this show, Dickson, because I could have given it to Twin, but I knew you would know this.
Well, there's a vector involved, the flea, and so we covered fleas a lot in our parasitic disease course.
All right, now we
transmit other things too.
Of course. Now we have a few concerning El Nino and La Nina.
Do you think that's a topic for this one or for the urban agriculture?
It's correcting us.
Well, I don't blame him for correcting us, that's for sure.
You can read one.
Boy, do we need to be corrected.
You can read Jody's.
Okay, so we already corrected this, but
but we have to read their...
Sure. Okay, so we get a lot of letters. This is from our other broadcast, by the way. Podcast, I should say, on urban agriculture. I mentioned... Wait.
No, we talked about...
Oh, we talked about malaria. That's right, and the spread of malaria throughout Ethiopia and in South America too. It's absolutely right.
Yeah,
absolutely.
You tilt the mic up a little. It's going. So it's rather directional. Thank you.
So it's Jody writes, "Hi, just a couple of corrections for when the discussion veered off into climatology, which was not my strong suit. I'm making an excuse already. La Nina is only the opposite case of El Nino, and has nothing to do with the Atlantic. There are, however, oscillations in the ocean and atmosphere in the Atlantic as well. That's true, such as the North Atlantic Oscillation. That's where I got confused. It's called NAO. It has been partially implicated in this cold winter in the U.S. and the warm, wet winter in Europe. El Nino/La Nina also have nothing to do with undersea volcanoes. Now, that's something that I didn't know because I had read somewhere that was the case. Okay, so I'm corrected at 2 levels now. At the most basic, they are caused by changes in the trade winds affecting the depth and warmth of the upper layers of the Pacific. When the trade winds flow in one direction, they expose cold currents, rich in nutrients, off the coast of South America and build up warmer water above Australia. The other way, the warm water covers up the cold South American currents, cutting off the important marine food sources and reduces the sea temps above Australia. As a New Zealander, I am very familiar with the Southern Oscillation Index, which is one of the main measures of El Nino intensity. As a resident of Finland, I am now learning of the effects of the NAO. Wikipedia, as always, has quite good descriptions of both, and as well as other regional phenomena. And as a side note, I would say that humans that cause... Now, I'll read this again, but I have to scroll down here, Vincent.
Do you know how to scroll?
I do. I know how to scroll. I'm scrolling as we speak. As a side note, I would say that human-caused climate change is about as controversial among climate scientists as HIV causing AIDS is among virologists. She means to say that
not controversial.
She's totally convinced that humans have had a major role in climate change, and so do I, of course, because I believe you guys. Thanks for the great podcasts. Even as a software developer, I enjoy the whole Twix series, although it is only TWiP that makes my skin crawl. Good. Terrific. You want to take the next one, or should I do?
You haven't finished. There's more.
Oh, regards, Jody.
Yes.
I won't read her last name.
There's a PSEP.
There's another PS.
PS.
Okay, PS. Has Vincent thought of giving all the regular Twix hosts the interview treatment like his recent podcast? I'm sure many are also interested in everyone else's backgrounds. Oh. Well.
Maybe I'll do that for number 300 in D.C., Dickson.
It's up to you.
Did you hear me?
I did.
I thought of doing that. We'll be all together. I can ask each of you.
This is all true.
Right?
Yeah, we can do that.
We could have a 3-hour episode.
We could do an autobiographical reveal.
Scott writes, gentlemen, as always, I listened with considerable interest to your discussion of malaria, having had it many times.
Oh, many times. Wow.
But when you got to the discussion of El Nino, that's where things went wrong.
That's when they went south, so to speak.
El Nino is not the result of oceanic volcanism, as you were speculating in the TWiP podcast of late March. The El Nino and La Nina pattern is a result of the interaction of persistent trade wind patterns and surface water temperatures in the Pacific Ocean. Dickson, you're fired. I've never received so many emails on a topic that we blew.
It's okay. No, there was one other, and that was the Norwalk virus origin.
Well, that was just a naming thing.
No, this is pretty bad, too. I agree with you there.
In normal years, the trade winds, which blow persistently from east to west, push surface waters in the Pacific toward the west. Current last 3 hours, trade wind activity in the Pacific can be seen dramatically in this graphic, and he gives a link to a pretty cool graphic. So, ocean water that is pushed to the west by the trade winds has to be replaced, and it's replaced by water rising from the depths along the west coast of South America. The water is extremely cold, and hence the penguin populations in the Galapagos, which are located on the equator, the last place you'd expect to see penguins, and hence the chilly climate of Lima on the tropical Peruvian coast. Penguins feed on herring and sardines, both cold water species, which flourish along the west coast of South America in the upwelling cold water. Cold water generates little evaporation, hence the coastal deserts. As the surface waters are pushed to the west, they accumulate around Indonesia, Malaysia, where they are pushed down into the depths. That deep, warm water is why that region is one of the most active thunderstorm regions in the world and the world's most active typhoon region, and with global warming becoming increasingly active.
There you go.
On occasion and for various reasons, the trade winds die down over a period of time, and the accumulated warm water in the western Pacific comes sloshing back to the east. Once it arrives in the eastern Pacific, the surface waters warm substantially, the sardine and herring fisheries die off, and the Peruvian fishermen starve. This usually happens around Christmas time, and hence the Peruvians give this phenomenon the name El Nino, referring to the Christ child in reference to Christmas. When this occurs, the warm water leads to increased precipitation, hence the flash floods in the Peruvian and Ecuadorian coastal deserts in El Nino years.
Got it.
When the trade winds increase above normal, the pushback to the west is increased and the opposite hydrologic effects occur. The cold water upwelling along the South American west coast is enhanced, and the fishing and the herring sardine fishery flourishes. This is La Nina. Here in Costa Rica, our climate is dominated by the El Nino-La Nina cycle, so we watch it very closely. Our worst drought on record was by far in the El Nino of 1984, the Great El Nino of 1984. So here is the current forecast for the El Nino 3.4 index for the next year, based on current climate models. A plus 0.5 or greater indicates an El Nino, and a minus 0.5 or greater indicates a La Nina. Close to the center line has been dubbed La Nada, the nothing. The red line is the most watched model. The cyan line is the model consensus. So he gives a map, which is a prediction. For the next year. So there it is, Dickson. You can see it.
I love it when you talk geologic stuff.
Right?
Yeah, no, I see it.
It's a prediction, and we're going to put this in the show notes.
You sure are.
I hope this clears things up for you.
Well, and everybody else that got misled by me, and I take full responsibility for this. I'm guilty as charged.
Regards from sunny and abnormally dry Costa Rica, 77 degrees as I write this. Scott is from Costa Rica. This is wonderful.
Thank you, Scott.
Dickson, our fans are really cool. They write stuff like this.
Not only that, they're knowledgeable.
They're knowledgeable, and he wrote a lot. He spent time on this.
Oh, listen, I have
We love all of our fans, right, Dickson?
We do. We have no ego problems with accepting the truth. How's that?
Is it your turn?
I guess.
Stacy is the next one, Dickson. Next time you need to fix your iPad.
I know we tried, but okay. Hi, Vincent Dickson. I came across this article on the IFLS website on Facebook and thought it was really interesting. I do confess that I don't listen to TWiP as often as I listen to TWiV. But would love to hear your thoughts about this study. I promise to listen to the episode. I can seem to download the article yet. Apologies for only sending the link. So what is that article? Again, an El Nino, La Nina article?
No. I can't seem to download the article. I know she wrote I can seem, but she meant I can't. The weather here in sunny Purbright is 16°C and partly cloudy. Quite a nice day in England, let's be honest.
Good.
Stacy works in arbovirus pathogenesis. She's a virologist. That's why she listens to
TWiV.
She sent a link to a Nature article, Dickson.
Yes.
Do you know the journal Nature?
I've heard of it. Which Nature? There's Nature Genetics, Nature this.
Tragocytosis by Entamoeba histolytica contributes to cell killing and tissue invasion. Does it make any sense to you, Dickson?
Read the article title again.
Tragocytosis.
Stop right there. What is that? Never heard of that before in my life.
You don't know what tragocytosis
You're right,
is?
and I don't think you do either. And you're looking at me.
No, the
like
internalization of fragments of living human cells is reminiscent of tragocytosis,
which is
observed between immune cells.
I see.
Amoebic tragocytosis differs because it results in cell death. You want to do a TWiP on this paper?
Sure.
It's Entamoeba histolytica.
Yep.
She wants to know what we think.
Well, after we read the paper, we'll let you know.
I think we should do it next time.
Fine.
What do you think?
Is it a recent paper?
Yeah.
Okay.
Our listeners only send recent things. It's April 2014.
I have had not heard that term before. I promise you.
Last one is from Robin.
Okay.
Homo sapiens is mostly an invasive species. The species is adapted to life in tropical climates. Any place that it needs clothes, it is an invasive species. Its natural migratory spread from Africa was through the now Middle East and South Asia to the Indonesian archipelago and thence to Australia. It did not need clothes for this.
Right.
He sends a link to Spencer Wells' The Journey of Man.
Right.
This is a playlist. Now, I'm wondering what we did to The Journey of Man. It's a series of wonderful videos.
Who produced it?
PBS.
PBS, okay.
The Journey of Man.
Because I've seen some very nice National Geographic presentations.
Spencer Wells. Who's Spencer Wells? Do you know?
I don't.
Well, it's basically the history of man, which is a nice... I think this is a counterpoint to the Jacob Bronowski series that we mentioned last time.
The Ascent of Man, yes.
So this is a link to support this idea.
Right.
Everywhere else, it is an invasive species needing clothing and shelter to survive.
Right.
This includes the migration to the New World, which passed through Beringia.
Indeed.
What?
Beringia. Yes, I know where that is.
Gnathostomata are the jaw-mouthed vertebrates, are distinguished from the agnatha, the jawless vertebrates, fish. The jaw is derived from the second branchial arch and lange. What? Okay.
Unlaga. It's called an unlaga.
Unlaga, thank you. The clade includes the sarcopterygia, the flesh-finned fish with muscles in their fins, which in turn cladistically extends to the tetrapoda. So gnathostomata are us.
And that's, there was a wonderful series. Yes, I do get it. There was a wonderful series on recently called Your Inner Fish.
Really?
Yeah, and it's by a really great paleontologist working out of the University of Chicago. And he was a wonderful host, and it was a 4 or 5 part series. And so he had your inner fish and your inner reptile and your inner amphibian. And he traced back through evolutionary history the origin of virtually every portion of us.
I'll put a link to that. And how's that?
I think that's a great idea because that really brings into focus the complexity of evolutionary history.
Yeah.
And, of course, the parasites that follow their chemical trails.
Oh, cool. So that's what we have to link
to. So parasites have to evolve with their hosts over time. Otherwise, they become parasites of something less evolved.
Well, or they may be extinguished.
Or they may be extinguished.
Same with viruses. They have to evolve with their hosts.
You know that there's a number of species of parasites. They follow the same patterns as the hosts. There's no difference.
Last paragraph of Robin's email. Thanks as usual for these most entertaining hours plus. A whole lot more interesting because the subjects are intellectually stimulating. Dr. Dickson's resolve to stay at the tiller till the stars guide him elsewhere is much appreciated.
Well, that's going to be that big ship in the sky eventually because I would never give this up. I'm having so much fun doing it, and I hope everybody out there realizes that. And I also hope you realize that I remain humbled by your collective wisdom. So your collective wisdom is what really makes this show legit.
Are you talking to me, Dickson? No, I know you're talking
to me.
Writers,
or you talking? No, I'm talking to
writers.
everybody because I'm just going to say. Remember my original saying on TWiP, which I will repeat now again on TWiP, and that is, trust science. Do not trust scientists. And I say that not meaning that every scientist is a dishonest person that would fake data and all that stuff. I don't mean that at all. I do mean that scientists that discover something and develop an hypothesis generally stick with that hypothesis regardless of the direction in which the data point them. For a long time, they will stay with that hypothesis and try to find out where they went wrong and try to do another experiment which verifies their original observations. And in many cases, they can't find that observation again. Another scientist will come along and says, we've done this experiment in a different way, and we found this rather than what you found. So, and then they get together and they fight like hell at meetings, and they become the worst of enemies. And then when they get older and they realize what science is all about, and that it's not about you and it's not about me, it's about the truth, they shake hands and say, you know what? Maybe because you were working with strain A and I was working with strain B, we were both right. Because what applies to A may not apply to B, and what applies to B might not apply to A. So I've seen that happen at meetings. I've actually seen that. And I'm sure you have too at meetings. You've gone to professional meetings where people have actually almost come to fistfights with each other because one believes in this and one believes in that. And remember, it's a belief. Science is not a belief system. Science is predicated on skepticism, followed by investigation.
You know what they say, Dickson?
What do they say, Vincent?
You can have your own ideas, but you can't have your own facts.
Bingo. 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 So anytime anybody starts to hear arguments based on beliefs, you just shut it off. Just turn that off. That's a religion. And religion is in another arena. We're not discussing religious ideas here. We're discussing what do we know about the natural world and our place in it. That's what these programs are really all about. What is the natural world all about, and what is our place in it? So that's basically
very dramatic, Dickson.
I feel I'm in a philosophical mood right now because
you should be on a TV show right now.
When I'm out looking at this window, I'm starting to see the high-rise buildings across the river now. But for a while there was no New Jersey. All right, so if you can't see it, is it not there any longer?
TWiP can be found at microbeworld.org/TWiP and also on iTunes. And we love your questions and comments, don't we, Dickson?
We love them.
Send them to TWiP at twiv.tv. Dickson Despommier is at Racaniello.org, medicalecology.com, and verticalfarm.com, and also urbanag.ws. Thank you, Dickson.
It's always a pleasure, as Alan Dove would say on another show.
Always a good time.
Always a good time. We've had a great time this time.
I'm Vincent Racaniello. You can find me at virology.ws. The music on TWiP at the beginning and the end is composed and performed by Ronald Jenkins. You can find more of his work at ronaldjenkins.com. In fact, the next Urban Ag, I should explain that we used one of his newer pieces. We have to explain why we used
Vivaldi's?
Vivaldi, and we forgot to do it on the last Urban Ag.
No, we actually did explain it, but we can do it again.
No, we didn't.
That's fine. We can do it again.
All right, ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.